In this video, we will go through how to use the pool tracking worksheet. The tracking worksheet can be used both before and after you buy your pooled screening library. You can work through the entire sheet before buying to calculate the functional titer in your cell line, optimal transduction conditions, and how many cells and plates you will need. Using the sheet, you can estimate the amount of lentiviral particles you will need. After you buy the pooled library, you can refine estimated values to actual values. You can find the pooled tracking worksheet on the CRISPR pooled library pages under the Resources tab. Once opened, you can see that values that you would put in are highlighted in yellow and the outputs in gray. The tracking worksheet can also produce a personalized protocol for your primary screen and library prep using our NGS library prep kit prior to NGS. The first step is to determine the optimal promoter for Cas9 expression in your cell type. It is important to do this because Cas9 expression levels have a significant effect on gene knockout efficiency. Next, we enter the optimal conditions for transduction, which are determined experimentally for the cell line that's going to be used in the screen. The conditions have to be compatible with a screen, and they should be conditions which give the best cell viability. For this example, here are values for a typical cell line. 0.5% FBS, 16-hour transduction, 10 micrograms per milliliter of a transduction enhancer such as polybrine, and 250 cells per millimeter squared. You can see that these numbers are being added to a personalized protocol in Part 6. You can use this for your primary screen. Part C is determining the functional titer in your cell line. In this example, we will be using an all-in-one GFP delivery control. However, if you have an EDIT-R two-part library, you will use a promoter-matched smart vector non-targeting control shRNA instead. You will know the exact titer of the pooled library as measured in HEC293T cells after you've received it, but for now, we will use an estimated value of 5.6 times 10 to the 8th transducing units per milliliter. You can use the GFP delivery controls to determine the functional titer in your cells. The titer of the EDIT-R all-in-one GFP delivery controls is reported on the certificate of analysis and was measured in HEC293T cells, which should be around 5.0 times 10 to the 7th transducing units per mil. It is important that you determine the functional titer in your cell line of interest experimentally because it will be different than that on the CFA. For now, we can use an estimated functional titer of 2.1 times 10 to the 7th transducing units per milliliter. Using these titer values, we are given a relative transduction efficiency of the cell line as 0.42. The sheet also gives us the relative functional titer of the pooled library in our cells of 2.35 times 10 to the 8th transducing units per milliliter. When you receive the library, you can go back and input the real titer of the pooled library instead of our estimate. Next, determine the antibiotic concentrations and durations needed to kill all non-transduced cells. For this example cell line, the conditions will be 10 micrograms per milliliter of blastocidin for 14 days and 7 micrograms per milliliter of pyrimycin for 5 days. This will vary depending on the cell type, so it is important to determine these values empirically. Next, we enter the fold representation that we want. Fold representation is the number of times an sgRNA is represented in a cell population. Generally recommend doing a minimal of 2 biological replicates. And we can look into the details of our pooled library to determine the number of sgRNAs. For our example library, Let's say we have a whole genome library with one pool of 76,000 sgRNAs, and we want to achieve a 200-fold representation. Next, we determine the optimal number of cells for transduction. With 76,000 sgRNAs and a 200-fold representation, the tool calculates the number of cells we will need with lentiviral integrations. We recommend an MOI of 0.3 so that around 22% of the cells will get one lentiviral integration and the remaining 78% will die in selection. To get enough cells with lentiviral integrations, the tool calculates how many cells we will need to transduce. Starting with this many cells at the density we determined previously and an effective surface area on 100 millimeter plates of 7,800 millimeters squared, we need 26 plates per sample. Since we want to do 2 biological replicates, that means we will need 52 plates per pool. Please keep in mind that if you're using a different library, these numbers will change. Now on to section H. The tool calculates how many transducing units we need and how many microliters of lentiviral particles that will be needed, taking into account the functional titer. For our 2 biological replicates, we will need 0.13 milliliters of lentiviral particles. If the functional titer of the cell line is lower, then we need more lentiviral particles. We estimated this value at the start of the worksheet, so it is important that you put your experimentally derived functional titer into the worksheet to ensure you are transducing your cells with an appropriate volume of virus. Before proceeding with the lentiviral sgRNA pooled library screen, confirm that you have sufficient lentiviral particles to perform the screen at the desired fold coverage given the relative transduction efficiency of your cell line and the number of biological replicates you wish to perform. Now we are on to the primary screen in section 6 of the tracking worksheet. In part C, we calculate the amounts of genomic DNA, the amount of PCR reactions, and NGS library prep kits needed for the screen. The tool has the number of cells with lentiviral integrations and multiplies it by the amount of DNA in nanograms expected in a human diploid cell. If you are working with a mouse or rat cell line or a polyploid human cell line, replace this value accordingly. The tool calculates the amount of nanograms of genomic DNA we need to maintain representation of each sgRNA. Then it calculates how many PCR reactions are needed per sample. In this example, if we have 4 samples, a reference sample and an experimental sample with 2 biological replicates, then we would need a total of 80 PCR reactions. The tool will then calculate how many NGS library prep kits will be needed for the screen. Lastly, for Illumina sequencing, please follow the manufacturer's instructions.